this video, we are going to look at restriction enzyme cloning in more detail. The key features of non-ambiguous type IIP restriction enzymes that make them useful for cloning is that they create double-stranded breaks in DNA. DNA binding is determined by a 4 to 8 base pair recognition sequence. DNA cleavage occurs between specific nucleotides within the recognition sequence. The result is a site-specific double-stranded break in a DNA molecule. Any particular enzyme generates characteristic double-stranded breaks. You can see 3 examples here. In each of these cases, the recognition sequence is 6 base pairs in length. Because type IIP enzymes function as homodimers, the recognition sequence is the same on each strand. With the strands running anti-parallel, the result is a palindromic sequence. Cleavage within this site is also sequence-specific. In the case of Hindi3, you can see that each monomer cuts at the same location within its side of the recognition sequence, between the 2 A's located at the 5' end of the binding site. The same pattern of palindromic recognition site and specific cleavage can be seen with the other examples. The location of the cleavage sites when Hindi3 cuts DNA results in a 5' overhang or protruding end. In contrast, when PST1 cuts its recognition sequence, the result is a 3' overhang. A generic shorthand term for either of these types of overhangs is sticky or sticky end. In contrast, notice that ECO-R5, while still requiring a 6 base pair recognition sequence, cleaves exactly in the middle of its sequence. The result is a blunt end. After we cut with restriction enzymes, we then use the enzyme ligase to create covalent bonds between 2 fragments. In order for ligase to function, it must religate compatible ends. Remember, a small number of type IIP enzymes contain some ambiguous nucleotides within their recognition sites. These enzymes are not generally useful in cloning. However, the majority of type IIP enzymes have non-ambiguous recognition sites, so the ends that they create are always the same. Hindi3 ends are always compatible with each other. Similarly, all ends created by PST1 are compatible with each other. And while it may seem obvious to some, Hindi3 ends and PST1 ends are not compatible with each other. 2 sticky ends are compatible with each other if they will form standard Watson-Crick base pairing prior to ligation. Blunt ends are different. In fact, all blunt ends are compatible with each other. Because the pre-ligation intermediate of a blunt end ligation cannot be stabilized by Watson-Crick base pairing, blunt end ligations are generally less efficient than sticky end ligations, but also more promiscuous. There are some notable details we need to add to this discussion of compatible ends. Let's start with isoschismers. Isoschismers are restriction enzymes that recognize the same sequence in DNA. As the names of our 2 examples, ASU-NH1 and NHE1, should indicate to you, each of these isoschismers was isolated from a different bacterial species. By definition, we see that ASU-NH1 and NHE1 both recognize the same 6 base pair sequence, GCT AGC. They also cleave between the first G and C from either direction, creating a 5' overhang. There is no way to tell these cleaved ends apart. They are 100% compatible. In comparison, we have neoschismers. Neoschismers are a subset of isoschismers. Neoschismers recognize the same recognition sequence but cut at a different location within that sequence. In our example, the isoschismers BMT1 and NHE1 are also neoschismers because, as you can see, they do not cleave at the same site within the recognition sequence. As a result, the ends they generate are not compatible. Some enzymes, which have different recognition sequences, can generate compatible ends after cleavage. 3 examples are shown in this table: NHE1, STY1, and XBA1. The internal 4 base pairs are the same for all of the enzymes, C, T, A, G. You can see the bases in positions 1 and 6 vary while maintaining the required palindrome. As a result, all 3 enzymes generate the same 5' protruding CTAG overhang. The compatible overhang allows fragments cut with any of these two enzymes to ligate. In green, you can see the 5' overhang generated by NHE1. In yellow, you can see the 5' overhang generated by XBA1. It is the 4 base pairs in the overhang that will hydrogen bond. After ligation, both NHE1 and XBA1 sites have been destroyed, so cleavage is no longer possible. Any two different blunt ends can be ligated together, so it can easily be the case that the ligated product will no longer be cleavable by the original enzymes. Here are the 5 basic steps that need to be completed in order to generate a molecular clone, which we discussed in detail in our video, The Plasmid Cloning Cycle. Let's walk through the specific example of cloning a fragment and coding GFP into the vector PUC19 so that we can get into more details. We are going to start our example by using a single restriction enzyme for the cloning procedure. By cutting our plasmid with a single restriction enzyme PST1, we open up the circle to create a linear molecule. The ends of the linear plasmid will both be byproducts of PST1 digestion. In another reaction, we will generate a fragment for insertion, also using PST1. In a third tube, our ligation reaction will be a mixture of both digested insert and vector, as well as ligase. If you are asking, won't all of the PST1 ends be compatible with each other? Then you're asking the right question, and the answer is yes, and this can cause problems. Possible results of this simple ligation reaction are listed here. Basically, any of the compatible PST1 ends are potential substrates for ligation. By far, the most common product that would be observed is a recircularized vector. That is because the ends of the vector are compatible and close to each other in physical space, a concept referred to as local concentration. Recircularized vector is often referred to as background colonies. Several strategies have been developed to combat this problem. The first of these is increasing the concentration of insert. Why would we do this? Invoking the chemistry part of biochemistry, we are increasing the availability of the insert as a substrate. Importantly, when we refer to the concentration, we do not mean mass concentration. We mean molar concentration. It is the number of molecules in the reaction that matter. In practical terms, we are actually referring to the molar ratios of the fragments being added to the reaction. In our general tips about molecular cloning, there is more information about molar ratios. When setting up a traditional ligation, we typically try to get the insert to vector molar ratio to be 2:1 or 3:1. A good practice is to actually perform a few different ratios, and be sure to have a vector with no insert control. A common and frustrating observation is that your vector with no insert control will have plenty of colonies, and this is of course expected, and a good result in that you know that your ligation conditions are working. But your vector plus insert has more colonies. Initially you think, great, I've got more colonies when I've added insert. But when you test some clones for successful insertion, you find only empty vector. This brings us back to the notion of local concentration. By adding more DNA in the form of insert, we have actually pushed the ends of the vector closer to each other, or increased their local concentration. On the other hand, there will be some colonies with the correct insert, but they will be lost in an ocean of background colonies. One tool we have at our disposal to distinguish clones that carry insert from those which do not is the blue-white screen. We introduced this technique in the cloning cycle video, but we'll review it here. Here is our PUC19 vector, with several restriction sites indicated within the multi-cloning site. If we linearize with PST1, we are cutting within a small protein called LAC-Z-alpha. LAC-Z-alpha is required to make a complete beta-galactosidase enzyme. When the chemical substrate XGAL is provided to functionally active LAC-Z, a blue byproduct is made. After cleavage at PST1 and religation, the result will be intact vector. We are attempting to insert a GFP gene into this PST1 site, which will also happen to disrupt the LAC-Z-alpha gene. The resulting colonies can be seen on this plate. When the plasmid has recircularized and LAC-Z-alpha is intact, we will see blue colonies as a result of beta-galactosidase activity. If instead we have succeeded in inserting our fragment into the PST1 site, the LAC-Z-alpha fragment is disrupted. Therefore, there will be no functional LAC-Z, and colonies will be white. Using blue-white screening, you will be able to find the coveted white colonies that are within that sea of blue. A completely alternative approach to using a screen is to minimize this background in the first place. And we have a few tools at our disposal to do this. Let's talk about phosphatase first. As its name suggests, phosphatase is an enzyme that will remove a phosphate. In this case, the 5' phosphate from an exposed end of DNA. The 5' phosphate is required for ligation to proceed to recreate the 5' to 3' phosphate sugar backbone of DNA. Once we have removed the 5' phosphate from the vector, we can see that ligation of the vector to itself will be impossible. Ligation will only be possible when the phosphate required for successful ligation is provided by the insert. If we were performing a cloning reaction using PST1 and planning on reducing our background colonies with phosphatase, we would proceed with the following steps. Digest your PUC19 vector with PST1. Treat the linearized vector with phosphatase. Remove or inactivate the phosphatase. Then proceed with ligation containing your appropriately prepared insert. Complete removal of phosphatase is essential. What will be the implication of phosphatase activity being carried over to your ligation? The 5' phosphate would be removed from your insert as well. If this happens, your ligation will not succeed. Therefore, it is extremely important to purify DNA away from residual phosphatase prior to proceeding with a ligation reaction. The other way to create vector ends that cannot ligate back together is to create ends that are not compatible with each other through the use of two restriction enzymes. The use of two enzymes in a cloning procedure is sometimes referred to as making a fragment or vector with double sticky ends. This is also referred to as directional cloning. Instead of linearizing PUC19 with just PST1, we can cut it with two restriction enzymes, let's say PST1 and BAMH1, both of which are found in the multi-cloning site. When performing this digest, we are not actually linearizing the vector, but are creating two fragments. One fragment is 14 base pairs long, and the other fragment is the remainder of the plasmid that we will use as vector. At the same time, we will need to create our insert so that it will have compatible PST and BAMH1 sites at either end. The only successful ligation products that could result in bacterial colonies are the desired vector ligated with the insert or the rare vector ligated to a second copy of vector, which could easily be eliminated when analyzing your clones. Let's again examine the ligation of our insert into our vector. Note that there is a useful benefit that comes from using two enzymes in this way to clone. The insert and vector can only align in one orientation. It is directional. In contrast, if we were using a single enzyme to clone, 50% of the recombinants would have the insert in one orientation and 50% would have the insert in the other orientation. When using two enzymes, which create two pairs of compatible ends, 100% of the insert will be in the orientation determined by these pairs of compatible ends. To review, we have just discussed how restriction enzymes are used to generate molecular clones. When necessary, we may need to use a single enzyme to process both the vector and the insert. As described above, this strategy will have a considerable amount of background colonies, which can make it tedious to find the desired clone. Three separate and complementary strategies can be applied to improve the outcome. First is blue-white screening. In this case, you are creating a phenotype that allows you to sort through the background. There are also a few enzymatic approaches available to reduce the formation of background colonies in the first place. One is the use of phosphatase to limit the ability of the vector to circularize. The alternative to phosphatase is to perform directional cloning using two restriction enzymes in your cloning procedure. Because all of these techniques have completely different mechanisms, any two or all three can be used at the same time in your cloning experiment. To learn more about molecular cloning, visit SnapGene.com.